I've been asked to give a talk on our recent research on how cancer cells metastasize. That is, how cells in a primary tumor leave the primary tumor and spread and form new tumor colonies in distant sites throughout the body. That is, the process of metastasis. This is more than just an academic issue, since metastases are responsible for about 90% of the deaths associated with cancer. Much of the research in my laboratory focuses on how carcinoma cells, which constitute about 80% of clinical cancer, undergo a profound change in their biology, where they change from an epithelial state, which is characteristic of the normal tissues from which these carcinomas originated, into a different state, a mesenchymal state, a connective tissue state. And this profound change in their biology imparts to the carcinoma cells the ability to invade nearby tissues, to enter into the bloodstream, to spread to distant sites, and there even to begin to initiate the formation of new colonies of tumor cells, that is, new metastases. One quite striking discovery that we made some years ago was that if one forces cells from an epithelial to mesenchymal state, one of the byproducts of that conversion is that often cancer cells enter into a state called the cancer stem cell state. And cancer stem cells are cells that have acquired the ability to seed new tumors. It turns out that most of the cells in a tumor, if you take them out of the tumor and put them in a mouse, will fail to seed a new tumor. But a minority of cells, the so-called cancer stem cells, have tumor initiating, tumor seeding ability, and thus are able to seed new tumors when they're implanted in a mouse. And such cells are also able to serve as the founders of new metastatic colonies should they spread to distant tissues in the mouse. We've only begun to learn how to target these cancer stem cells, which indeed are quite dangerous because they exhibit an elevated resistance to most existing forms of therapy. And as a consequence, research in a number of laboratories has begun to attempt to develop drugs that specifically target the cancer stem cells, the minority of cells in tumors, rather than the majority of non-stem cells in the tumors, which are more responsive, more sensitive to killing by existing chemotherapeutics or radiotherapy.
And what are some of the other points that you'll be highlighting in your talk?
In addition to the description of how cancer cells spread, I will also be describing the fact that we've begun to understand why certain breast cancers become aggressive and others don't. This turns out to reflect the fact that certain cancer stem cells have the proclivity, the ability to spontaneously go from a benign epithelial state into an aggressive mesenchymal state, whereas other cancer stem cells lack that ability. And as a consequence, for example, luminal cancers of the breast lack the ability to generate new cancer stem cells, whereas basal carcinomas of the breast, which are quite nasty often, readily generate or spawn new cancer stem cells, which, as I've said before, can be quite dangerous. The work that I describe is still far away from actual clinical translation. One might say, some might say, well, if you can't translate it into clinical application, then why do it? But the fact of the matter is that the research of this sort is discovering that part of the reason that existing clinical therapies have failed is attributable to the existence of these cancer stem cells, whose existence was only realized over the last 5, 6, 7 years, and which represent a hidden enemy that now has come into light and now represents an obvious and highly attractive target for therapy. That doesn't mean that, in fact, this will be immediately translatable. I was involved in discovering a protein in 1981, which 25 years later became useful in treating breast cancer, that is, through the antibody Herceptin. So the discovery of some basic fundamental attributes of cancer cells is not instantaneously translatable into clinical applications. The take-home message from my talk will be that there's far more to determining the behavior of cancer cells, and thus tumors, than the sequencing of their DNA, because these conversions of cells from a non-cancer stem cell state to a cancer stem cell state don't involve changes in the sequence of DNA molecules, but rather a rewiring of the signaling circuitry within the cancer cells that does not involve shifts in the structure of the DNA. Currently, there's much interest and focus on sequencing the genomes of cancer cells, but it's my conviction that such sequencing efforts reveal only part of the aberrant behavior of cancer cells, and one will need to study these non-DNA-based changes in cells, which will in turn require an entirely new technology in terms of evaluating the data, that is a new form of bioinformatics, to be able to integrate information on how cancer cells' DNA and mutations in the DNA and the non-genetic, non-DNA-based changes in cells conspire with one another to generate the final aggressive state of many carcinoma cells.